Awesome. Can you hear me? Cool. Okay. This is the thing. Okay, cool. So I'm going to be talking about the role of the heart, and I think you've had a lot of kind of introduction for interoception, so I'm actually happy that I could remove the slide on the importance of interoception. But I'm going to be looking at the role of the heart in maybe a topic that hasn't been discussed that much in this kind of summer school and conference, and that's the perception of time. So right now I'm standing here and I'm giving this talk, and although I know there's a lot of familiar faces, but I'm still a bit nervous, and I can feel my heart is beating much faster in my chest. And the way I experience time right now feels very different to me than how it felt when I was listening to the talk and sitting down. And that reflects that our experience time does not reflect time on the clock. It is highly subjective and elastic. And I hope that the example that I've given you shows that our own internal state, our emotional state. But anyways, while they do it, I can finish my introduction. So our internal state, our emotional state plays a really important role in how we experience time. But in order to get to the actual emotions and phenomenology of time, I am starting from a very low level. So I'm starting from the perception of very, very short moments and how they are affected by each heartbeat. And in this talk, I'll present you some very recent EEG correlates of that effect. So we studied the role of the heart by looking at the effect of phasic fluctuations in the heartbeat. So I think you talked a lot about the systole and diastole. And I just wanted to point out, I mean, if you were at the summer school, I think Ezra was giving a workshop, but I like to point out the study because I think it's really nice. So in that study, they were presenting participants with very weak somatosensory stimuli, either during the systole or the diastole, and participants were seeing if they detect the stimuli or not, and they found that participants were less likely to detect the stimuli during systole, and the important thing is that was related to the suppression of this P300 component in the somatosensory evoked responses. And the important thing, why I like to point out the study, is that the effect was not early. So it's not that systole just adds noise to the system, but it seems that it's more to do with some kind of a top-down attenuation of sensory processing for stimuli that coincides with the systole, with cardiac signaling. And that basically made me think, well, if cardiac phases affect sensory processing, would they also affect how we perceive the duration of that stimuli? So here we use a temporal bisection task, and essentially what you have in a temporal bisection task is participants do a learning phase when they learn to distinguish a short and long reference, and then in the testing phase we present them with the durations in the middle. And on each trial, they basically say whether the stimulus duration was more like the short or more like the long one. And in our behavioral study, we did 2 experiments. In one, we presented a visual stimulus or an auditory beep. And experiment 2, we presented faces with happy or fearful expressions. And we were recording ECG, which allowed us to time lock these durations to either systole or diastole. So the durations are quite short, so it's like 200 to 400 milliseconds because we wanted them to neatly fit into the cardiac phases. And essentially, when you plot participants' data, what you get is the proportion of long responses against the test durations. And here I'm showing you an example, participant 1. And the important thing that you can get from this graph is the point of subjective equality. And ideally, we want it to be around 300 milliseconds because that was our bisection duration, which could be either long or short. And if the PSC is smaller, that means that the durations were overestimated. And if it's bigger, it means the durations were underestimated. So this blue line is the diastole. And if we plot the systole, you can see this divergence between the blue and the red. And this is just an example participant. And for that, we see the systole contracted perceived durations relative to diastole. So we did see a lot of variability. So not everybody shows this pattern. But in that study, we did find a significant effect. And the interesting thing in the experiment, too, is that we also were asking participants to look at the faces, but we also asked participants for arousal ratings. So here, when I say arousal, I mean like emotional intensity and like threat. And if we actually took arousal ratings into account, we find that this cardiac effect is not always there. So anyways, I'll continue. So the arousal effect was there so the cardiac effect tended to be there when participants gave lower arousal ratings. And as experience arousal increased, that cardiac effect actually started to disappear. I think it's a really interesting effect. I'm not sure. I'm going to come back to it in a second. I'm not quite sure exactly what it means yet, but I'll come back to it at the end of the talk. But essentially, what we find for at least a low arousing situation is that systole tends to contract time, where diastole tends to expand it. So our next step was very simple. Let's put an EEG cap on people and see what are the actual EEG correlates of that effect that could predict this temporal contraction. So we did a visual temporal bisection with 41 participants. Everything was the same. So here, I'm just going to show you the evoked responses for the visual, kind of the visual areas, so occipital channels and parietal channels. And I'm plotting, this is only when participants were experiencing the 300 millisecond stimuli, so the stimuli that were at the bisection. And for the kind of responses during the occipital channels, you can see very clearly the evoked responses that you should be seeing. And the solid line is the diastole and the dashed line is the systole. Here, you don't see an effect of cardiac phase. However, if you look at the parietal channels, so at the 300 milliseconds, so the stimulus started at 0, and then it ended at 300. And once it ended, you do start to see this divergence, and this is a significant time window of this effect, where systole tends to suppress what I called here a late positive component relative to diastole. And what is interesting and what is important, I think, is that this EEG effect was correlated with the behavioral effect. So here, participants who tended to show a systolic suppression in the EEG response were also the ones who tended to show a contraction of time by systole. So basically, what we showed is that the suppression of the LPC was related to duration contraction. And just in general, from the timing theories, I was wondering if this could be a support for coding efficiency accounts of timing that basically say that instead of like having some kind of a central clock in our brain, the duration and temporal kind of qualities are just intrinsic to sensory processing. So usually in the audible paradigms, if you present participants with a standard stimulus, the perceived duration gets shorter and the neural response gets shorter. And once you present an audible, suddenly you have a stronger neural response, but participants also perceive the audible as lasting longer. And indeed, there's been studies that tie the kind of sensory, the P300 component with like enhanced P300 would be temporal elongation. However, one thing is we don't find the effect on early sensory processing. So it does tend to be some kind of like a later component, but I think it still shows that it has to be something with how the sensory stimulus is processed that kind of relates to how we experience the time, the duration. And essentially what we find is that also for time, we need to take time as an embodied experience. So it comes from this interaction between our visceral senses and the brain. And the thing we find is that during the systole durations, these very short moments tend to be contracted, whereas during diastole, they tend to be expanded. Now, usually things last longer than just a single heartbeat. And if you are having your normal heartbeat and you're experiencing these longer durations, then from this, these effects should cancel each other out. So actually, when we're in a normal situation, probably we don't see any temporal distortions because these effects cancel each other out. It's more when one starts to impact more than the other, then we should see actual temporal distortions for longer durations. And indeed, when we have a lower heart rate, we would get more inference from diastole, and we should see dilation of time. Whereas when the heart rate gets higher, we'll get more inference from systole, and we should see the contracted perception of time. And indeed, one of the studies have also shown that. So one thing that I just want to point out before I finish is that I think the effect of emotion and arousal is really important. Again, here I'm talking more about emotional intensity. So here we're kind of looking at the situation of low arousal. And in low arousal, stimuli associated with our own heartbeat may be top-down attenuated because they're treated as noise. Because when we're in a low arousal situation, we don't really need to pay attention too much about what our heart is doing. However, if we go to a more higher arousal situation, what happens is that now the homeostatic regulation of our body becomes important. So suddenly these cardiac signals should get a higher precision, they would have a higher impact on our perception. So something else might be happening. I'm not sure what. Should we see like an enhancement of components? So currently we are collecting data for the EEG of that experiment. So maybe if this happens next year, I'll present these results. But I just want to leave you with this quote from Murakami in Kafka on the Shore, where he said, "Time extends, then contracts, all in tune with the stirrings of the heart." And thank you so much for listening. Yeah, thanks.
Thanks for a great talk. Do we have questions? Diego has a question.
Hi, Irena. Nice
Hi.
talk. So what you were saying at the end, like when you have a longer cardiac cycle, you have longer time for it, time to expand or contract. But did you actually check that in your data? So there was a kind of interaction effect.
Yeah, so in the analysis where we look at the effects, we do put in our model the actual like IB length, so how much there was like a heart deceleration, acceleration. We didn't find in that case an effect. So there's a cardiac phase effect, and it's independent from the deceleration, acceleration of the heart. But in that study, they did find it. So it's kind of maybe these effects would not really happen just within one heartbeat. Maybe you need kind of acceleration over multiple heartbeats to actually see the frequency effects. I'm not quite sure about that yet. Or like maybe you need a much stronger acceleration or deceleration that you typically see in the experiments that we do, where it's kind of a low arousal situation.
Because I have in mind, like, so considering with our hearts are different size.
Sorry, say again?
Our hearts have different size.
Yeah.
So what is high heart rate for you might not be for me. And
yeah, but I do think that it's more to do with your heart rate.
Yeah, that's why I was thinking. So it's not easy to test that. So that's why I was asking.
Yeah.
If you do, how will be done by.
Yeah.
Thank you.
Will you be quick? Okay.
Thank you very much for your talk. It was very interesting. I would like you, if you can ask you to speculate, if you think that the effect that you're finding on time distortion can drive other effects as an emotion, is likely arousal that is commonly driving these time distortions and also, for instance, emotion cycle effects.
Sorry, can you repeat this?
Do you think that these effects, cardiac timing effects, are driven by arousal, both your time distortion, other people results on emotions, or rather that your time distortion effect that can actually drive the effects on emotions?
Oh, so you mean like if the emotional effects are actually driven by temporal distortions?
Right, or they're both driven by arousal.
Oh, I see. Yeah, well, yeah, so kind of mean that systole is already kind of high arousal signal. Yeah, I don't know. I think it would be interesting to finish the actual emotional EEG to see if now in the fearful we'll see the same effect. So I almost feel like under high arousal diastole would become more like the systole, right? Because now it's kind of, we're pushing everything into the high arousal situation, but we'll see once we get it. But that's a really good question. Thank you.